Hello, everyone. Thank you for joining us today to hear another exciting seminar or webinar that we're hosting at INAMIN. Before we begin, I would like to just mention a few rules that we have during the webinar. So first of all, during the whole webinar, all the participants will be muted. And if you have any questions to our today's speaker, please type them into the Q&A, not in the chat, but this Q&A box. And once there, I'll be happy to read them after the presentation. If at some point your colleague or colleagues are not able to attend today's webinar, it's okay, don't worry. We'll be able to share the recording of the webinar with you probably later today or tomorrow. This recording as well as other recordings from all the events we've hosted, they are stored on our YouTube channel. So today we'll be talking about exploration of ultra-large or gigascale chemical spaces. And you probably, if you know INAMIN, you probably heard about INAMIN Real. Yeah, I'll just remind you what it is. It's not. I hope you probably not can see my screen at the moment. Okay, so that's you see my screen. And if you go to inamin.net, we have multiple products here, and one of them is INAMIN Real. And basically, INAMIN Real stands for a collection, a huge collection. We can call them zillions of compounds that can be synthesized on demand. They're not in stock. They're actually synthesized on demand since we know how to synthesize them and we have all the building blocks. And this is really a powerful instrument for those involved in heat exploration, heat to lead, and to some extent into compound collection enhancement programs, where people can find novel molecules and basically get new heats fast. The INAMIN Real concept has been on the market for a while, and as you can see, it's pretty trustful among our customers who published a lot of publications, a lot of papers citing INAMIN Real, and as you can see, the number of citations has been increasing in time. And yeah, we have different ways to represent or to show you what INAMIN Real compounds, and those are some kind of known names like INAMIN Real Space, which includes the Real database as a subset and some others. And yeah, if we go to INAMIN Real Space, you probably know that we constantly grow in it. And we have multiple ways to explore it. We have the enumerated version, we have the Synthon-Based versions that are really popular since screening 80 billion molecules is kind of too much. And by the way, we are about to release an update to our INAMIN Real Space, as well as other INAMIN Real Space and database related products next week. So please keep tuned. So yeah, so the space is growing, and obviously the demand for the tools is needed. And today I would like actually to tell you that we have a really great opportunity to explore a really nice way for searching and exploring the INAMIN Real Space. And today we have our speaker, Dr. Enrique Herrero, who will be presenting their approach to screen ultra-large chemical spaces. Few words about our today's presenter. So he's graduated from Polytechnic University of Catalonia. It's probably in Barcelona, I believe. And then after a few positions, including one at Intel, he actually co-founded a company called Pharmaxellera. And yeah, I think it's a really great company with a number of useful tools that help drive your research. So with that, I would like to give a stage to Enrique, yeah, please go ahead, and looking forward to really exciting presentation.
Thank you, Yuri. Let me start sharing the screen. Okay.
Yes, we can see the
Great.
screen. Yep.
Perfect. So thank you for the introduction. I'm going to start the presentation with just a simple question, that is, are these two molecules similar? At a first sight, you might say that these two molecules are very different, but however, these two molecules are both known A2G inhibitors. However, if we look at these molecules from the bioactive conformation, we start seeing that there are some common patterns in the molecular properties that they have. And if we look at the hydrophobic profile of the molecule, then we see that really the hydrophobic profile of both molecules is very similar. At Pharmacelera, we are using this hydrophobic profile of the molecules for different computational chemistry tasks, specifically for virtual screening, scaffold hopping, SAR analysis, and also for creating predictive models. The advantages of using this type of fields are mainly that you can encounter more diverse molecules in your virtual screening campaigns and do it in a more accurate way than with simpler descriptors. As Yuri was mentioning, the size of the accessible chemical spaces has grown exponentially during the last years, being Enamine one of the first companies to start exploring and offering this ultra-large chemical space to the clients, reaching 70 billions, or as Yuri was saying, now even more. And this raised a question when these libraries started growing of how can we mine efficiently this type of libraries without sacrificing the quality of the results that we are obtaining. How can we deal with the amount of data that is associated with this type of libraries? And also important, are the compounds that we are exploring synthesizable, or they are intellectual chimeras that we are not going to be able to test? So in this talk, I'm going to talk about different alternatives that exist nowadays to explore ultra-large chemical libraries. Then I'm going to introduce our methods, which are exaScreen and exaDock, and present you the results of a validation study that we have recently published. And finally, I'm going to talk a little bit about how you can access this technology in your drug discovery projects. So exploring this type of ultra-large libraries, if we are using a simple Morgan fingerprint, exploring 1 million compounds takes around 3 seconds. But if we start increasing the size of the library to up to 1 hour, can take up to 1 hour. Something that is more costly, but still something that is doable. However, the accuracy of this type of descriptors is not suitable for all applications. If we want to use descriptors as the ones that Pharmacelera has, like the hydrophobic profile, exploring 1 million compounds in a single machine can take up to 11 hours. And if we aim to explore billions of compounds, this might take up to 16 months to complete. Of course, if we are a company that we have many resources and access to the cloud, we can use high-performance computing infrastructure or the cloud services to accelerate this and reduce from 16 months to a few hours if possible. But still, this is going to be at a very high cost, which we might not want or not capable of to afford. Therefore, we need to think of alternative solutions to solve the exploration of these chemical spaces. Different alternatives that are available nowadays. Brute force, as we have said, has the problem of the computational cost that it has. Active learning is a method that has been used by many people because it has the main advantage that it can be directly applied to existing tools that existed from the past, and consists in adding a machine learning model to sample the chemical space, and with just 1% of the chemical space exploration, you can obtain the same results as brute force. However, 1% of 70 billion still represents a significant computational cost, and therefore we thought that it would be interesting to find other alternatives. Generative models are another type of method to increase the size of the chemical space that is explored by the usage of generative artificial intelligence methods. The main issue that these methods have is the synthesability of the molecules that have been proposed. There are methods to assess the synthesability, but however, the cost of synthesizing these molecules and the time that is required to have them for experimental testing is unknown. And this is something that sometimes in our drug discovery projects we cannot afford. Synthon-based approaches, basically what they do is use the building blocks that are used to create these ultra-large libraries instead of the fully enumerated libraries, and are much more scalable. Therefore, this is the method that we have used with our tools, exaScreen and exaDock. So in this talk, I'm going to talk about the methods that are behind exaScreen and exaDock. Then I'm going to show you the validation that we have performed for both of the tools to assess that the quality of the results is comparable to what we can obtain with brute force. And finally, I'm going to cover a little bit how these tools can be used or accessed for your drug discovery projects. The work that I'm presenting today was published last April at the Journal of Chemical Information and Modeling. So if you are interested in knowing more details, please refer to this publication to know more. The tools that we are going to present are exaScreen, which is a ligand-based virtual screening tool, and exaDock, which is a receptor-based virtual screening tool. The general idea of Synthon-based tools, as I was saying, is that instead of comparing full molecules in a ligand-based approach, what we are doing is using the building blocks, like the Lego bricks that are used to create these ultra-large chemical libraries, in order to perform the search. And instead of looking for the similarity of all the compounds, just looking at the similarity of the building blocks. In terms of computational cost, this has a significant advantage because if the number of building blocks that is used to create these ultra-large libraries increases, the computational cost and size of the library increases exponentially, and also the computational cost for brute force methods. However, Synthon-based methods, the computational cost increases linearly and is, first of all, much lower computational cost, but also it will scale better in the future when these libraries are going to continue to grow. The first of the two methods that I'm going to present today is exaScreen, which is the ligand-based approach. The idea behind this tool is that what we are doing is instead of using the full ligand, we are partitioning the ligand in several fragments, and then we are computing the hydrophobic profile that we have shown before that allows you to retrieve more chemical diversity. And we are also generating this field of interaction for all the synthons of the different chemical libraries that we want to explore. Once we have the hydrophobic profile of the synthons, we compare all the synthons against each of the reference fragments, and we select the best ones for each of the reference fragments. Finally, what we do is that we combine the best synthons for each reference fragment, but not in an arbitrary way, but taking into consideration the reactions that are possible for these building blocks. Therefore, enumerating only molecules that belong to the enamine real chemical space, and that you can purchase easily and at a reasonable cost and time. In the same way, exaDock is a tool that works with synthons, but instead of being a ligand-based approach, is a structure-based approach in this case, after partitioning the reference structure, what we are doing is that we are joining one of the fragments with all the syntons of the library, and then we are performing a restrained docking to find which building blocks or syntons are more suitable for this region of the binding pocket. Afterwards, we do the same for the other region of the binding pocket, and finally, we select the best syntons for both regions and enumerate the most promising molecules using only the allowed reactions. Finally, we perform an unrestrained docking, and we get a list of purchasable compounds. The questions that arise after presenting these questions are two. Basically, what would be the computational cost of these methods? And second is, can we get the same performance that we get with brute force? So basically, in this study, what we did is that we pick 11 diverse targets from the DUT-enhanced database, which include reference structures and also active ligands and decoys to assess the performance of virtual screening campaigns. What we have done is, on one hand, select building blocks to enumerate this chemical space. And in the case of brute force, we work with the fully enumerated molecules. In the case of exaScreen and exaDock, we work with the building blocks. The general idea is to see if we are able to retrieve the same hits by the brute force methods and the Synthon-based methods, as we are going to see. First of all, we are going to focus in the validation of exaScreen, our ligand-based approach. Here we can see the results of the virtual screening campaigns with both methods. In pink, we can see the results for exaScreen, and in orange, we can see the results of the brute force approach. As you can see, for the different receptors that we evaluated, the performance of the two tools is very similar. In some cases, we got a slightly better performance for the brute force approach, that is PharmScreen. And here we can see with different gradients the different enrichment factors for the virtual screening campaign. So initially, we can see that more or less we can obtain similar results in terms of the overlap of the hits that we obtain in the top 0.1% enrichment up to the top 0.5% enrichment. We can see that except a couple of cases, in most of the sets, both the brute force and the exaScreen methods are capable of retrieving the same hits. Here I'm going to show you just an example of the chemical space that is being enumerated with the different methods. This is for the RXR receptor, and this is the reference structure that we use. First of all, what we need to do is to partition this molecule and then select the chemical space that we are enumerating. Here we can see in red the reference structure that we have used for this virtual screening, and in yellow we can see all the hits that we want to retrieve. Here, just for comparing the chemical space that is enumerated by different methods, we have also enumerated ligands using simple 2D descriptors, and we can see that if we pick the top 1% of the ranking, we are enumerating molecules that are very close to the reference structure. If we look at the molecules, the active molecules that we identify in this chemical space, we can see that there are chemical structures that are very similar to the reference. On the other hand, if we see the chemical space that is enumerated with the brute force method or also with exaScreen, first interesting thing is that for both tools, the chemical space is very similar. Second interesting aspect is that it is a chemical space that is broader and that also covers the region where other active compounds are. if we look at the hits that are identified in this extended chemical space, we can see that there are hits with much more diversity and therefore more interesting from the intellectual property perspective. Entering into the details of other specific sets, we have seen that there are some sets where the performance of exaScreen is lower than the performance of FarmScreen, the brute force approach. So we have analyzed the reasons behind the differences in performance. In this specific case, what we saw is that if we analyze the fields of interaction of the syntons and of the complete molecule, here we have the reference structure, and below we have different hits that we want to identify. We can see that for one of the two syntons, the field of interaction of the fragments of the hits is different from the reference synton, and therefore we are not capable of identifying these hits. On the other hand, if we look at the field of the complete molecule, since the part that is different is smaller in comparison with the size of the full molecule, this difference is not affecting the selection of the molecule, and we are capable of retrieving these hits with the brute force approach. In the opposite way, there are cases in which exaScreen performs better than brute force. At the beginning, our goal was basically to obtain the same results as brute force, and we were surprised of getting better results, but then we understood the reason behind that, and this is due to the better conformational adaptation of the different ligands to the reference structure. Here we can see two examples where the performance of exaScreen is better than in brute force. We can see the overlay of the hits on top of the reference structure using the Synthon-Based approach and using the brute force approach. And we can see, of course, that the conformer that is selected in exaScreen is much better, and this is because the flexibility of the syntons is much lower than the flexibility of the full compound. We have performed an analysis increasing the number of conformers that we use in the brute force approach, and we could be able to retrieve better results. But however, here we have a trade-off in terms of the computational cost, because we do not want to have a very high number of conformers when performing this analysis. So therefore, we have seen that in general, for exaScreen, for the ligand-based approach, the performance is very similar for brute force and the Synthon-based approach, but in some cases there are advantages of using the brute force approach. In other cases, there is an advantage of using the Synthon-based approach. And the second question that we were considering is the computational cost. So here we can get the numbers that we got, and this is for the enum in real space of 70 billion. With brute force, this would require 1.5 petabytes of storage to store all the parameters and conformers of this chemical space, and would take in a single machine 95 years to complete. On the other hand, with exaScreen, it requires only a library of around 6 gigabytes of storage, and we can run this simulation in 7 hours in a machine of 48 threads. So this is 100,000 times faster and 260,000 times less storage, which I think, considering the performance that is similar, is a significant advantage. And not only that is a significant advantage, but enables the exploration of these ultra-large chemical spaces if you don't have very high computing resources. The second validation that we have performed is the validation of exaDock, our structure-based virtual screening approach. In this case, we can see that the difference in performance are higher, and we can see that exaDock in some of the sets is performing worse than the brute force approach. In this case, the brute force approach that we have used is glide-SP. In terms of the overlap between the hits that are retrieved by exaDock and glide, we can see also that there are more differences in terms of the hits that are retrieved. So if we analyze the reasons behind this difference in performance, one interesting case is map K2. In this case, we analyzed the docking scores of the synthons that belong to active structures and also the synthons from the decoys. And here we can see the results when restraining the part of the ligand that is inside the pocket of the protein and the results of the synthons when we are, sorry, it's the opposite. This is when we are restraining the inside of the pocket, and here when we are restraining the solvent exposed area. So interestingly, what we can see is that in the when we are finding synthons in the area that is inside the pocket, we are capable of detecting the synthons from active molecules. But however, when we are looking for the synthons of active molecules in the solvent exposed area, the active synthons are having worse results in the docking than some of the decoys. However, we have to say here that in this case, the decoys are not true decoys. So we don't really know if the decoys that we are selecting here would be worse binders or not, because at the end, what we are forcing is the activity in the region that is inside the pocket. The region that is more solvent exposed maybe does not have that high impact in the activity of the compound. However, well, for the performance analysis that we did, the results were worse in this specific case. There are, as in the case of exaScreen, some cases where exaDock is better than the brute force method. And here we can see some of the scenarios. And this is similar to the case that we encountered with exaScreen, where we were able to better adapt the conformer to the referent structure. So in this case, we can see here the poses that are generated using exaDock with the restrained docking, that is taking into consideration the pose of the reference structure. If we use a brute force approach that is not restrained but by the reference structure, what we encounter for this specific receptor is that the docking tool was not probably generating the correct pose for some of the active compounds. As you can see here, it was positioning some of the active compounds in a different way. And therefore, this could be an explanation of why the brute force approach was not capable of retrieving those hits. Another interesting aspect of this study is the overlap of the different active compounds that have been identified by the two methods. On one hand, we have receptors like ABL1 that has a solvent exposed area where the overlap is very low. And once again, this is related to the fact that in one of the regions of the molecule is less restrained in the position that the ligand can have because it's solvent exposed. On the other hand, if we have a more constrained binding site like the H1 receptor, we can see that the hits that are found by the two methods are very similar, and this is because there's less flexibility in how to position the ligand in the pocket. Overall, in terms of computational cost, here we can see that in case that we want to use the brute force approach with the full enamine real space, it would take around 468 years to complete in a 48 threads workstation. The usage of exaDock allows to complete this virtual screening in just 78 hours. So this is 50,000 times faster and 100,000 times less storage. Overall, in this case, the performance is not as good as the brute force, but however, this is also an enabler because basically it would be impossible for someone to perform the brute force docking for these ultra-large libraries. So in this way, we can obtain results in 78 hours. So this completes the part of the validation of exaScreen and exaDock. I wanted to spend some time also in explaining how to leverage your own internal chemical spaces. So something that we encountered when talking with some clients was the question of, okay, so we can use commercial libraries, but we would like to use also, in addition to enamine real, our own building blocks and reactions that we have internally, our internal chemistry. So basically, we have also developed a tool that is called exaScreen Intra that allows you to pick all the building blocks that you have in-house, all the reaction lists, and then convert them in a virtual library that you can use to perform a search that retrieves only molecules that you have the capability to synthesize. As an example, we can see here how the information would look like. So what we would use with exaScreen Intra would be the building blocks that you would provide, and then a specific reaction smears, in this case for the triazole formation. So the tool, what it does is it is converting these building blocks into these specific syntons, and therefore this syntons is the information that is used by exaScreen or exaDock to perform the chemical space exploration. Finally, last two slides, I would like to focus in how to visualize the results and access the technology. First of all, we have also, in addition to exaScreen and exaDock, we have a visualization tool that is called MolExplore that can be used for the post-processing of the results provided by these two tools. First of all, it allows you to better understand the reasons behind the selection of these different compounds. Here we have an example of the reference structure that we were seeing before, and one of the active ligands that exaScreen was capable of identifying. If we look at the selection of and the 2D structures, we might think if they really are similar, but using MolExplore, we can visualize the overlay of the 3D structure. We can also visualize the hydrophobic profile of the molecules, and then we can realize that the two molecules are very similar, and therefore they could be active both in the same receptor. MolExplore not only allows you to perform this analysis, but also allows you to filter the compounds based on different properties like molecular weight, log P, cluster the molecules in order to select representative from different chemical families, and basically allow you to streamline the selection of compounds that you want to purchase to enamine, for example, using this tool. In terms of how exaScreen and exaDock is executed, basically we have a flexible access to the technology. At Pharmacelera, we provide the software to be executed in the client's infrastructure, and also we provide CAD services in order to perform the usage of these tools for a given project that you have, and you don't have to worry about the installation of the licensing of the tools. As of today, exaScreen is available both as software license and CAD services. exaDock is right now available only with services, but will be eventually also available as a standalone tool that you can run in your infrastructure. As a summary, I would like to say that, well, the accessible chemical spaces like enamine real are increasing exponentially. And as Yuri was mentioning, this represents an opportunity to improve the drug discovery process with newer and more diverse hits. At Pharmacelera, we have a unique 3D molecular modeling technology that is validated with publication and success cases and enables the identification of novel and diverse hits, and therefore helps you in the identification of new intellectual property and the ideation of new chemical structures in your drug discovery projects. exaScreen and exaDock open the doors to use these 3D descriptors in ultra-large chemical libraries because they are capable of reducing the computational time 100,000 times and the storage 260,000 times. Finally, we have a flexible access to the technology through software license or services projects, and we would like to hear from you if you think this technology would be interesting for you. So thank you very much for your attention. Now I would leave time for questions if you have.
Yeah, thank you. Thank you, Enrique, for this really exciting presentation. So we have we have a few questions, and remember, please don't forget them to put your questions into Q&A box. So yeah, so this technology is actually based on synthons of fragments. So are these strategies you presented compatible with fragment-based drug design? And if so, how are they adapted for those like formats?
Yeah, so basically the general idea is that since we are working already with fragments when performing these virtual screening campaigns, this means that this technology can also be used for scaffold hopping or fragment growth, in the sense that we can use a fixed fragment that either has been retrieved in a fragment-based drug discovery project, or also that we want to perform a scaffold hopping search, and then try to search for different scaffolds in the other part of the molecule using the same methodology.
Thanks, Rick. So that's again more about the technology. The question is more about the technology. How do you handle the issue of conformational flexibility in both ligands and proteins during the screening process?
So basically, what we are doing is that we are generating conformers for all the synthons. Typically, for the reference structures and the protein, we rely on the crystal structures that would be available, but otherwise we enable also the usage of relaxed conformers or conformers obtained from docking. And then what we do is sample this conformational space in order to be able to adapt the ligands to better match the hydrophobic profile of the reference structure.
Thanks. How do you envision Synthon-based strategies evolving with the rise of AI or ML-driven de novo design and generative chemistry approaches?
Yeah, so I think that generative AI is a complementary technology that especially I see it more suited for later stages of the drug discovery process, where basically you can afford more the additional cost and time that is required to synthesize the compounds. However, in early stages of the drug discovery project, the fact of using libraries like EnamInReal, what allows you is to control the cost and time that is required to synthesize these compounds, and therefore and test them experimentally earlier, and also you have the capability of testing more compounds and getting more insights on the SAR of the given receptor you are working. Therefore, we think that both technologies are complementary and will coexist in the future.
Yeah, thanks, Henrik. So is there a way to implement consensus docking or different core functions?
Yes, so it is possible to apply different scoring functions to the technology, and at the end the methodology is agnostic of the scoring function. So yes.
Thanks. Question is related to the actually combination of synthons, how you combine them. So how do you combine the synthons? Is there any bed torsion, filtered, etc.?
Yes, so it is very important that we ensure when we are joining the syntons that we are getting conformers that are realistic. So therefore, our tool also includes a way of ensuring that the conformers of the molecules that we are enumerating are reasonable. Yes.
Thanks. Yeah, now we have about 80 billion molecules, and if we go to 800 billion molecules, how is there? I mean, your technology, will this be pretty? I mean, the screening time, will this expand linearly, like 10 times,
Mm-hmm.
or
Mm-hmm.
have you tried
Yeah, that's
exploring larger spaces?
Yeah, that's a great question, and in fact, what we have seen is that basically Synthon-based approaches are the only method that will scale properly in the future, because since it's based not on the size of the chemical library but on the number of building blocks that are used to create this library. If the size of the library increases exponentially, the computational cost for tools like exaScreen or exaDock will only increase linearly. And therefore, the bigger the library is going to be, the bigger the advantage of Synthon-based methods will be.
Okay, thanks. And for the current library of 70 billion molecules, what are the minimal requirements to run if a user wants to run them on their infrastructure?
Mm-hmm. So we have been running this in a single desktop PC of 32 threads, and it takes around 7 hours. The code is parallelized, so if you have machines with higher number of threads, you can even run this in 1 hour. And the same way, if you have smaller machines, it will take a little bit more, but also will be in the range of few hours, let's say.
And speaking of machines, do we need to have a powerful cores, or rather just we have it and rather we need to have more threads?
Mm-hmm. Yeah, so of course, the more powerful cores that we have, the faster it will be the calculation. But since the code is parallelized, the more threads that we have will scale more or less proportionally. So if you have twice as many threads, it will run twice faster.
Thank you. Are exaScreen and exaDock models parameterizable?
So I don't understand what would be the parameterizable way, but in some sense, what we have in the tool is different ways of adjusting the search in terms of the requirements that the client might have in terms of, for example, filters of certain molecular properties, filters of certain substructures that we want to have present in the output molecules, and also filters on specific pharmacophoric points we want to have in certain positions. These type of filters allow you to perform a more fine-grained search in these ultra-large chemical spaces to adapt to the requirements of your project.
So I think there is a question related to the preparation of syntons. So how do you handle molecular charges, charge states with Synthon-based approaches?
Mm-hmm. Yeah, so basically what we are doing is the libraries like Enamine Real are already prepared at physiological pH, and we are calculating the protonation of the syntons. Finally, once we have the full molecule, we are correcting the protonation in the linker area, which is the area that might be affected by changes while moving from a synthon to a fully enumerated molecule.
Yeah, thank you. And there is one question related to probably to us, to Enamine. So quantity, size of libraries doesn't mean quality of compounds in accessible libraries. How can you trim and improve the quality of your Synthon libraries? Well, that's what we've been doing for many years now. We actually, before putting compounds into the library via Synthons or via enumerated format, we validate the compatibility of the corresponding reagents and keep only those compounds which have reagents with pretty high synthesis success rate. So that's why we are able to support pretty high delivery of compounds within a 3-4 week time, and that's what the users of the real space like about the product. Yeah. Thank you. Maybe one more question I have. So since we're kind of about to have this new release coming soon, and you receive the Synthons, how fast can you prepare the Synthons once you receive them, basically, that ready to screen and further used in exaScreen, exaDock?
Yeah, so this is something that is very fast. We have an automated way of preparing the libraries, but however, from the user perspective, is something that you don't have to worry because we are providing the fully prepared libraries. We have a partnership with Enamin to get access to this information, and you will only have to download the prepared library and worry about performing the virtual screening campaign.
Thank you. We don't have any more questions at the moment. Yeah, if you still have questions, please do not hesitate to contact me or contact Enric directly, and we would be happy to answer them to you. Yeah, with that, I would like to thank you, Enric, for this really exciting presentation, exciting, I think, Q&A session. Thank you, everyone, for joining us today. And yeah, I would like to close today's seminar. And please, I remind you that please follow us on our LinkedIn or Facebook or other social networks to find out about more webinars that we'll have in the future. Well, thank you, everyone. Have a good one. Bye.